the transcription of the viral genome is not used, right? Then the machinery continues and actually transcribe the SART gene, the C-SART gene as well. But then the C-SART gene also contains multiple polyadenylation sites, and it just happens that in thousands of years ago, it terminates right there, missing the last domain, basically missing the last exon. And we know now that I think it's just one exon. I don't think it's more than one exon, but an exon. But that exon, that very last exon, is really important for the actual regulation part of the SART protein. Without that, there's no regulation. Okay, so that's, yes sir?
You're saying it gets all the function without the regulation? Or yes. A lot of the function?
That's a lot of the function without the regulation is exactly what I'm talking about. So as you can see, studying SART, that big idea is already huge, right? People had no idea. So that idea of viral oncogenes for tumor viruses were acquired that way. And that's why practically all of the viral oncogenes have a cellular proto-oncogene version. So that's the big idea. Yes, ma'am?
If that polyadenylation site is in the middle of our SART gene, the cellular SART gene, how come we're not making that protein ourselves?
It might be made, but it's better regulated. How is that? Right. So for the SART gene, it's always synthesized that way. Whereas and that's a fair question. So we do also make this truncated version as well, but it's well regulated, meaning that the amounts may be less, or it could be that we have it, but it might be a mistake. Could be that polyadenylation site is there. It's a mistake, and that whenever a truncated SART protein is produced, it gets ubiquitinated and then degraded. That part I actually don't know, right? That part I don't know. But so in our cell, that is well regulated. Now a virus, when it infects us, what happens to the viral proteins? They get produced en masse, a huge amount, versus a normal cell. Even if, let's say, you put a V-SARC gene into our cell, right? Your cell would not automatically express the V-SARC gene. It has to be controlled. Whereas during a viral infection, right, the V-SARC gene is produced in very high amounts. So that's the best I can answer that. And I don't know. I'm assuming that people know exactly what's going on, but I just don't know that much, okay, at that detail. Okay, but the general idea is that viral oncogenes, actually practically all of them have a cellular version of it. Okay, so that's again one big idea. All right, so now let's look at the V-SARC versus C-SARC protein in closer and see what is the difference, right? What is that missing exon? What does that do? Okay, and then get to actually more big ideas. Well, the second big idea is that SARC is a kinase. So that's one of the very first kinase that is identified. And so what is a kinase? Kinase is an enzyme that carries out phosphorylation to regulate protein function, right? And during in the signal transduction pathways that regulate the cell to carry out cell proliferation, right? Phosphorylation is very well used. Right? The MAP kinase cascade, for instance, just about everything gets phosphorylated to either be turned on or turned off, including the receptor itself, right? So kinase activity is huge. So again, stemming from the study of SARC, people understand about kinase activity. So that's the second big idea, okay? The third big idea, believe it or not, is originally when people eat, again, on SARC, people study SARC and they said, oh, kinases. Originally, people thought that kinase activity only turns on an enzyme. And phosphatase activity only turns off, which is incorrect. Through the study of both D-SARC and C-SARC, not just one or the other, both we find out that kinases can turn on or turn off an enzyme. And phosphatase can turn on or turn off an enzyme. So the point is that SARC is a kinase itself, but it gets turned on and turned off also by kinases and phosphatases. Okay? So in order to regulate a kinase, phosphorylation and dephosphorylation also does that, even though the kinase itself carry out phosphorylation. Right? So again, phosphorylation, dephosphorylation occurs everywhere. So did everybody understand this? So this is another big idea, and this is actually pretty recent. So Dr. Rao's study identified Rao's sarcoma virus. Only reason why I remember this date is in 1912 is because that's the exact same year that I went to college, and the college where I went was also established in 1912. So that's the only reason why I understand. I remember. So anyway, and the date doesn't matter, but all the way right more than a century ago, but yet we don't know about the idea of kinases not only can turn on a protein enzymatic activity, but also can turn off. We didn't even know that until probably the 1980s. We're talking about 70 years later before we understand this. Again, at first people thought that kinases turn on, phosphatases turn off, and that's a good assumption, right? Why would you have those two activities if you don't want one to generally turn on and one to generally turn off? Well, that idea was disbanded, was proven wrong by studying SARC. And here's the idea. C-SARC first. C-SARC. If this were C-SARC right here, I would draw, let's say, this picture right here. Draw C-SARC that way, where this is right here is the catalytic site, the site that it binds to a substrate so that it can bind to a substrate that it can regulate enzymatic activity, right? So that's C-SARC right there, C-SARC protein. Right? In order to expose that catalytic site, so again, that's active sites. How about active sites? Because so proteins that aren't enzymes can also have active sites, right? So we're talking in general. It doesn't have to be the catalytic site. It could be an active site. In order to activate C-SARC, here's what has to happen. First of all, that the tail here, we just call this a tail. And this, by the way, is the last exon. Okay. So that tail acts as an inhibitory domain that covers the active site. And how do you get this tail to open up? Conformational change. How do you carry out conformational change? Phosphorylation. So you actually add the phosphate group right there, causing steric hindrance, and it causes the tail to flip open. And the active site is there. Okay. However, the active site is exposed doesn't mean that C-SARC is active. So now it's exposed because of that phosphate group. In order to further activate C-SARC, C-SARC has to be hyperphosphorylated, meaning that an additional phosphate group has to be added. So it's sorry about this. So hyperphosphorylated. phosphorylated. Okay. Okay. So that's how this is. And that's how you can activate it. Okay, let me give me a second. Let me think. It's been a long time since I did this. Let me work it out myself first. Give me a second here. It's been a really long time. Figuring out if I tell you the right thing. Oh, okay. Slightly different. That's right. Again, slightly different. So let's get back to this. I'll keep this in. So, sorry. Okay. So the idea is actually you want to get the enzyme in this conformation, this phosphorylation conformation right here. Okay? How do you get to that phosphorylation conformation? You get that by phosphorylating here first. That's right. Okay? Okay, causing steric conformation, and then the tail gets kicked out. Okay? Yeah, this is it.
So you still have phosphorylation on the tail?
Not yet. Okay, so you want that. So this is active. You want it to get to that conformation. That conformation where it's phosphorylated at site 1. Let's say at site 1. You might have phosphorylation site 2, but site 2 is unphosphorylated, right? So to be active, you want this. You want site 1 to be phosphorylated only, right? So far so good. Slightly different from what I said. Okay. Now, so if you phosphorylate site 1, site 2 is not phosphorylated, but still, it's open. Okay, conformational change is open, active. Now, in order, sometimes when you regulate protein function, if it's in that form, but you want to keep the same protein and you don't want to completely inactivate it or degrade it, right? So you want to be able to control the phosphorylation and activation and activation and inactivation very rapidly. Okay, so basically what you want is, again, phosphorylation at site 1, that's active. That means that the tail is open, right? By phosphorylation here, the tail is open. Then if you want the tail to close without actually undergo dephosphorylation, what you do is you phosphorylate at the second site. When you have a hyperphosphorylated form, you actually inactivate the Sark again. Okay, but because of the conformation. So actually, I shouldn't draw this. Is this to inactivate, and this is to activate, right? So then once it's formed and you activate, in order to activate and inactivate, you switch back and forth from in these two versions right here, right? Yes, ma'am.
So phosphorylating site 2 is going to make the tail close.
Close, right? Because again, conformational change. Okay, yes, sir.
You call both of them hyperphosphorylated or just the inactivated one?
The inactive, there are two versions of inactive C-Sark protein. One is the completely unactivated, right? This is inactive. And the hyperphosphorylated version. The singly phosphorylated version is the only active version. Okay? You notice phosphorylation inactivates. So it turns phosphorylation activate here but inactivate here. Right? That's another big idea that study Sark did. Not only that, but how do you get this? Right, this is site 1 and site 2. How do you get this inactive version to be activated again? Dephosphorylation. Dephosphorylation going the other way. Another big idea is that people used to think that dephosphorylation only turned off an enzyme, but in this case it's turned on. Right? So looking at Sark, the C-Sark alone, you can see that both direction has been proven wrong in that kinases can both turn on and turn off a protein, and dephosphorylation can do the same thing, turn on or turn off, depending on what? Depending on the protein itself, depending on the enzyme, and depending on conditions and a lot. Right? So sorry about the rocky beginning. Like I said, I haven't thought about this in like 20 years. But does that give you an idea of how Sark is regulated? And so therefore, the point is that that last exon is really, really important, right, for the regulation of activation, inactivation. Yes, sir?
Can you go straight from zero phosphorylation to hyperphosphorylation? Phosphorylation?
First of all, what does it mean by hyperphosphorylation? Which one do you phosphorylate first?
The first one?
Yeah.
Do you have to do the first one?
What happened if you phosphorylate the second one?
Nothing.
It doesn't exist because that's in the conformation where the second site is hidden.
Okay.
Right? It's only exposed when the tail is exposed. So number one. So it's physically impossible. But number two is why? Why would you want to do that? Right? So, yeah. And again, I'm not picking on you. I'm just trying to give you a very logical reason why yes or no, and you could have answered that by yourself.
Yeah.
Okay? We're not done because we haven't said anything about it. But you can guess what happens is, why do you want to do this? You want to do this is because, like I said, Sark. Actually, Sark has multiple active sites. This is not one, but for the most part, Sark carries out phosphorylation of a number of substrates, and there are dozens, literally dozens of them. Right? They all somewhat has to do with the cell proliferation, and basically, the Sark, the active Sark protein, right, feeds into this RAS. MAP kinase cascade, and those of you who are in cancer biology, just a reminder, those of you who haven't taken it or are not in it, then it's then don't worry about it. But this right here, when we say cell proliferation, right? Remember that it's not just one pathway that leads to there. It's different pathways, right? It leads to increased translation. It leads to decreased apoptosis or no apoptosis at all, right? Right. It leads to cell motility. Motility, roughly, right? And it leads to entering cell cycle by producing CDK and cyclin. Right? So one signal alone. Now this is not all because it has something to do with contact inhibition as well. So right, so contact inhibition. It has to do with, I'll just write with contact inhibition, right? Right. So, and Sark actually feeds into active, let's say C Sark feeds into that. So when you turn on Sark, when you activate it, right, you're gonna be activating all of these things that actually are very important, and you might need a few things here and there, but that can be accommodated. You know, no apoptosis, no contact inhibition, just about everything is there. Just that one pathway. In Bio 121, you remember the hardwiring, and one signal can activate a bunch of different pathways. This is what that is, right? So that's why activating Sark is important, and you only need one signal again, so that when you want the cell to undergo cell proliferation, you don't have to gather all the material. You just want to say yes once, and then you're done. Okay? All right. And again, the uptake of nutrients and everything else is necessary, but minor enough that you might be able to continue without signals. Okay? So far so good. And therefore, it has to be well regulated. I'm going to keep this. I'm going to erase this right here. So that's C Sark. Now, what happened to V Sark? Right? What happens when you don't have that tail? Well, first of all, you still have to phosphorylate the site 1. So this V Sark still has to be upregulated or activated. But once that site 1 is turned on, there's no way to turn it off, right? Once it's turned on, because you want that active form, there's no way to turn it off by phosphorylating what? There's nothing to phosphorylate. So it's only dangerous so if there's a way to keep the Sark protein from being activated, then you don't have a problem. The problem is that usually the Sark is phosphorylated. Basically, it has to do with anchorage dependence. So if you have anchorage, that means that you have the focal adhesion or focal contact. Sark protein can be recruited there. So chances are, unless you have a system where you don't have anchorage at all, and normal cells have to have anchorage, right? So this is actually dampening. So for cancer cells already, this might not be a problem, but for normal cells, this is a problem. Does that make sense? Because normal cells have to anchor. Cancer cells don't have to anchor. So if normal cells have to anchor, then chances are Sark will get recruited anyway and get phosphorylated, whether it's the B Sark or the C Sark version. So this is why, right? And if it's recruited and activated, there's no way to turn it off. So therefore, this activity goes up, and everything else, cell proliferation occurs uncontrollably, because this is already on. There's no way to turn it off. Right? So now it's uncontrollable. At the beginning, the very first step of activating it is controlled, right? Because it has to be phosphorylated. But from here on, everything else is uncontrolled. Yes, ma'am.
I feel like I'm asking the same question, but site 1 can't just be dephosphorylated without that tail?
Usually, it can be dephosphorylated, but usually not, right? Usually not. And yes, it does have something to do with the tail, yes.
Is the tail used in proteases? Like, is it an attachment point of proteases where you have inactivation from the secondary? Okay, when you have hyperphosphorylation, you have the tail coming back up with that changed conformation. So is that the conformation the proteases are looking for to break it down? Because I'm wondering why.
There's no breakdown. There's no proteases involved. I'm not sure. There's no protease involvement here. There's no digestion of any kind.
Okay. I'm just wondering why there isn't any degradation at any level.
No. But there's no degradation. These are healthy proteins. So there's no degradation of protein.
In host cells that are undergoing cell proliferation, at a certain point they need to stop, and so you're going to want to get rid of the C-sarc.
That's different regulation when things get turned off, and then you have to go through the cell cycle, and then once the cell goes through the cell cycle, there is a point where that is regulated. But it's not at this point. This is at the upstream of the cell cycle. This is way upstream of the cell cycle. This is like the cell is in G0 and it wants to get back into Z1. G1 is this, which is completely different from the M phase.
Okay.
So no, don't get that out of your head. No protease activity. Don't worry about that at this point. Right? This is way upstream. Okay. So the removal of this is again the missing exon that is found in the VSARC gene that is in the Rosarcoma virus. That's why this is missing. I thought that's what you were asking. So this is not digested. It's just not made to begin with. Okay. So that's why when a Rosarcoma virus, right, infecting let's say an avian fibroblast, that greatly increases the chance of carcinogenesis. The question is why, if it's so effective, why doesn't the Rosarcoma virus infect human cells? Let's say. Yes, sir.
So the only difference between the inactive and the active VSARC protein is the phosphate group.
Yes and no. The difference is that not only phosphate group, but whether or not the active site is blocked. It's phosphate group, but it's blocked. It's phosphate group, but it's not blocked. Here's a phosphate group, but it's not blocked. So the whole point is whether you have a single phosphorylation or two phosphorylation events. That's the point.
VSARC protein?
You didn't say VSARC, you just said for the SARC protein. So
I
for
meant
VSARC,
protein.
yeah, VSARC is activation, but that's the same as the C-SARC, right? If you phosphorylate site 1, if you just have phosphorylation on site 1, has nothing to do with dephosphorylation. There's no dephosphorylation. As long as site 1 is phosphorylated, right? Then in VSARC, there's no site 2, there's only site 1.
That's what I mean. So the active site is never blocked.
Right.
So why does it need to be phosphorylated?
Because that's how you activate the activity.
Okay, so that's good.
Right. That's why I said that. Just by having the site unblocked doesn't mean it's active. It has to be phosphorylated to be active. Why do you have to phosphorylate anything? Why do you have to phosphorylate anything? Maybe this would help. Because what you just asked is just memorization, right? I don't want you to memorize. Why do you have to phosphorylate in site 1?
Conformational change.
90% of the time you are correct in saying that conformational change will regulate protein function. So phosphorylation here will get the protein in a certain conformation. Not only to get the tail to come off of the active site, but also to get the active site being in the proper conformation. So maybe I should draw something like the square here versus the circle here. Right?
If I see how it looks like when it's active, the VSARC protein, I will.
Right. So active would be circle, and inactive would be a square. Let's say. Right. Actually, that's not even true in this case. This is still a circle. It's just blocked. Whereas if you don't have phosphate group, then it's a square. So unphosphorylated, you have to get it into this conformation, also without the tail. And then even if it's in that conformation but it's blocked, then no. So two things have to happen to activate SARC. To get SARC to be active, one is the right conformation of the active site, and the other one is no tail. How's that? That's better, right? Okay. I didn't know where you were going, but I see why you have that the hesitation. Hope that explains it. Okay. All right. Okay. Now I lost my train of thought there. There was oh yeah, a question. So this is this virus, right? Only infects chicken avian cells. Why wouldn't Rosarcoma virus infect human? Be real careful. Okay, be real careful. Think about everything that we talk about, okay, in this class, and then I'll add a little bit to it. So if you drink, right, like I said, if you drink a test tube full of Rosarcoma virus, you will not develop a tumor automatically. There's a possibility, yes, but more than likely you won't. Why is that? It has to do with two words: susceptibility and permissibility. Your cells are not susceptible because they don't have the receptors, right? I had to figure this out. No, when I first started doing this work in the late 1980s, a long time ago, almost 30 years ago, I was told that, oh yeah, don't worry about it. It doesn't bother you. And I thought, why wouldn't it? It's so bad for the fibroblast, but you don't get affected. It took me forever to find out that it's very simple. Your cells are not susceptible. Your cells are permissive, though. Your cells are permissive, right? So this is the extra layer. So that's why we separate susceptibility and permissibility. Is that the reason why Rosarcoma virus will not infect and cause problems because your cells are not susceptible. But what happens if you take the viral genome and you use biotechnology and insert into your cells? You bet you that you're going to develop some kind of a cancer-like phenotype, or even develop tumors. Yes, that. So that's the difference between susceptibility and permissibility, and all that is the permissibility part is that truncated means a shorter version. That truncated version of the V-Sarc is so powerful that if it gets made and be activated in your cell, in any cell, right, that automatically allows your cell to be susceptible, unless of course the cell loses the ability to undergo cell proliferation by having a lot of these, like terminally differentiated cells. Most of them don't undergo cell proliferation. Chances are those cells, even if you do this, will not develop a tumor. But if you do this to let's say an adult stem cell, you bet you that you're going to develop some kind of a tumor, right? Okay. Yeah, you don't want to ask. Too shy, or I answered it.
It's just a really far off idea.
Go ahead.
I was just thinking, can you have like a possible co-infection that would make the cells susceptible?
Sure, yeah, sure. I don't know if this is. I don't know if this is in reality it happens, but think about the parvo